when we talk about a major global disease, we usually assume the villain is clearly defined, you know?
Right, like it has a specific pathogen, a distinct name, a neat little box.
Exactly. We assume it sits on its own isolated branch of the evolutionary tree.
Well, yeah, I mean, it provides the sense of clinical comfort, right, to have biological threats categorized perfectly in their proper taxonomic boxes.
But what if I told you that the textbook villain of a massive global illness doesn't actually exist?
Which is still just wild to me every time we talk about it.
It's crazy. By modern genomic standards, the bacteria genus known as Shigella, the infamous cause of bacillary dysentery, isn't a separate genus at all.
No, it's really not.
It is literally just our common, everyday gut resident E. coli wearing an evolutionary disguise.
Yeah, it is the absolute definition of a taxonomic lie, and the global medical community simply agreed to just keep telling it.
So today, we are going deep into Shigella. For this deep dive, we're going to unpack how this bacterium uses a microscopic toolkit to hijack human cells, why it remains a massive public health threat, and basically why taxonomists let this biological fiction persist just for the sake of saving lives.
It's a fascinating story.
It really is. And to start, I want you to imagine a standard, everyday family car that represents your normal symbiotic E. coli.
Right, the harmless stuff.
Right. Now imagine that on at least 7 separate occasions in history, totally different mechanics independently bolted on heavy armor and weaponry to turn that mundane family car into a literal battle tank.
That analogy captures the genomic reality perfectly, because Shigella does not represent a single continuous evolutionary family tree.
It's more like a functional category, right?
Exactly. On multiple independent occasions, different ancestral strains of E. coli picked up this specific piece of genetic hardware. It's a massive virulence plasmid known as PINV.
And plasmids are like mobile DNA molecules.
Right. And this particular one contained the complete genetic blueprint for cellular invasion.
So these different E. coli strains essentially just downloaded a weapons schematic.
They downloaded the weaponry, and this is the crazy part, they optimized for stealth.
Oh, wow. How do they do that?
They actually silenced several of their own native genes, like the ones that build flagella for swimming.
Because the immune system would spot those.
Exactly. Those structures are really easily flagged by the human immune system.
Yeah.
So any E. coli that convergently evolved to acquire this specific invasion plasmid and adapted to use it just gets grouped under the clinical label Shigella.
So it's not a biological species at all. It's a job description.
That's a great way to put it, yes.
But, you know, to grasp the devastation this job inflicts, we really need to trace the origin of that clinical label. The sources point back to a terrifying public health emergency in Japan right around 1897.
Yeah, and the scale of that epidemic is just difficult to overstate. In a single year, Japan saw roughly 90,000 cases of severe dysentery.
90,000 in 1 year.
Yeah, resulting in over 20,000 deaths. It was horrific. And in the center of this crisis was a young bacteriologist named Kiyoshi Shiga.
And he was working under someone pretty famous, wasn't he?
He was. He worked in the lab of Kitasato Shibasaburo, who was this towering figure who had already helped isolate the plague bacillus. So Shiga was tasked with finding this invisible killer that was just sweeping the nation.
But he didn't have modern genomic sequencing back then?
No, not at all. He had to rely on foundational germ theory. So he isolated a rod-shaped bacillus from the stool of dysentery patients. But finding a bacterium in the gut isn't proof of causation, you know?
Right, because the gut is basically a jungle. It's full of bacteria.
Exactly. So his methodology to prove causation was brilliant. He utilized agglutination.
Which is blood clumping.
Yes. Shiga took blood serum from patients who were recovering from the disease, and he exposed it to the bacillus he had cultured, and the bacteria clumped together.
Because the recovering patients' immune systems had generated specific antibodies against that exact pathogen.
You got it. The clumping proved the human immune system recognized this specific bacterium as the invader. And furthermore, he only found this bacillus in the sick patients, never in the healthy controls.
So he published these findings in 1898.
He did, naming it Bacillus dysenteriae, and then later in 1919, the scientific community renamed the genus Shigella to honor his work.
Which makes sense. And the specific strain he identified, Shigella dysentery type 1, is still the most virulent member of the group today, right?
It is, yeah. It's uniquely capable of producing lethal amounts of the Shiga toxin.
Okay, I want to pause on the word dysentery for a second, because there is this widespread misconception about what that actually means.
Oh, definitely.
A lot of people, they treat dysentery as just an antiquated term for a bad stomach bug or severe diarrhea, like Oregon Trail stuff.
Right. Just, oh, you have an upset stomach.
Yeah, but clinically, dysentery is a completely different mechanism of disease.
The distinction really comes down to tissue integrity. If you consider how cholera operates, for instance.
Okay, cholera.
Vibrio cholerae remains in the lumen of the small intestine. It releases a toxin that acts on the cellular machinery to basically actively pump chloride ions and water out of the tissue.
So it's just flushing everything out.
Right. The result is massive, large volume, painless, watery diarrhea. In the medical field, we call this secretory diarrhea.
Meaning the cellular architecture of the gut is fundamentally intact, it's just leaking fluid?
Exactly. But Shigella operates on the complete opposite end of the spectrum.
It bypasses the small intestine entirely and
Right.
targets the colon, right?
Yes. And rather than just secreting a toxin from the surface, it physically invades the tissue.
Which sounds brutal.
It is. The clinical presentation reflects that violent invasion. It induces a massive inflammatory response. Patients get high fever, severe abdominal cramping, and tenesmus.
Tenesmus, that's the constant urge to go, even when there's nothing left.
Yes, the relentless, painful urge to pass stool, because the bacteria are actively necrotizing or destroying the mucosal lining of the colon. So the resulting stools are small volume and streaked with blood and mucus.
Which is the hallmark of invasive inflammatory diarrhea.
Exactly.
So how does a modified E. coli pull off this tissue-destroying heist?
The invasion toolkit is fascinating, and it apparently all starts with an incredibly low infectious dose.
It really does.
Yeah.
Because most enteric pathogens face a severe numbers problem. The human stomach is this incredibly harsh, acidic environment designed to neutralize threats.
Right. It's an acid bath.
Yeah. So to establish a successful infection with salmonella or cholera, you typically need to ingest hundreds of thousands or even millions of organisms.
Because the vast majority just perish in the stomach.
Right. Only a small fraction survive to reach the intestines.
But the human challenge studies from the University of Maryland showed Shigella plays by entirely different rules.
Oh, yeah. In the 1960s and 80s, researchers gave healthy volunteers carefully measured doses of the bacteria, and the data proved it only takes 10 to 200 individual organisms to cause a violent clinical infection.
Just 10 to 200? That is absurdly low. It's because of its extreme tolerance to gastric acid. Almost the entire swallowed dose passes through the stomach unharmed.
And that low infectious dose completely alters the epidemiology of the disease, doesn't it?
Completely. Because it removes the need for an incubation period in a contaminated food source.
Right. You don't need a potato salad sitting in the sun to multiply the bacteria into the millions.
No, you don't. It can spread through microscopic trace amounts of fecal-oral contact.
Which makes it highly transmissible in environments where direct human-to-human contact is dense and sanitation is compromised.
Exactly. That's why we see explosive outbreaks in daycare centers where toddlers are interacting closely, or in refugee camps lacking clean water infrastructure.
And the sources mention it's heavily tracked as a sexually transmitted infection among men who have sex with men, too.
Yes, it just needs a direct ride to the next host. It's incredibly efficient.
Okay, so once those 10 to 200 bacteria reach the colon, they face a solid wall of epithelial cells lining the gut. They can't just drill through the front door, can they?
No, they can't. So they look for a back door. Specifically, they look for the M cells.
M cells.
Yeah, microfold cells. They are specialized immune sampling cells, integrated directly into the intestinal lining. Their whole biological purpose is to constantly sample the environment of the gut lumen.
So they swallow tiny particles and transport them across the epithelial barrier.
Right, they hand them off to the immune system's macrophages in the underlying tissue. It's a surveillance mechanism.
And Shigella willingly gets swallowed by these M cells. It treats the host's surveillance system as a free shuttle service across the heavily guarded border.
That is exactly what it does.
But wait, once it gets dropped off on the other side, it's immediately engulfed by a macrophage. I'm stuck on something here. If the macrophage's literal job is to digest and destroy bacteria, how does Shigella survive being swallowed?
It seems like it's playing right into the immune system's hands.
This sounds like suicide.
It does, until the PINV plasmid activates. Once it's trapped inside the macrophage's phagosome, which is basically the cellular stomach, Shigella deploys a type 3 secretion system.
A type 3 secretion system?
Yeah, you can visualize this as a microscopic molecular syringe. It physically pierces the membrane of the macrophage from the inside and injects a payload of bacterial effector proteins.
And the primary one is called IPAB, right? Directly into the immune cell's cytoplasm.
Yes, IPAB. And IPAB triggers a biological self-destruct sequence.
Wow.
It initiates a highly specific, violently inflammatory form of programmed cell death known as pyroptosis.
Pyroptosis, as opposed to regular apoptosis.
Exactly. Normal cellular death, apoptosis, is quiet and contained. The cell just sort of packs itself up. Chiroptosis is a fiery explosion.
So the macrophage just erupts?
It ruptures, releasing the Shigella into the underlying tissue, but it also floods the surrounding area with a massive wave of inflammatory cytokines, most notably interleukin-1 beta.
Oh, wow. Think about the implications of that mechanism. The bloody stool, the intense cramping, the tissue damage that isn't just the bacteria chewing through the colon.
Not at all.
It is the host's own immune system overreacting to that cytokine flood. The host's defenses are causing massive collateral damage.
The pathology of dysentery is fundamentally an immune assault gone haywire.
It's like the ultimate Trojan horse. But instead of sneaking soldiers into Troy to quietly open the gates, the soldiers intentionally trigger the city's own defense armory to blow up the city walls. They ranganize the host's alarms.
That's a perfect way to look at it. And with the tissue now severely inflamed and the physical barrier compromised, Shigella has access to the basolateral side, the unprotected back door of the intestinal epithelial cells.
So it invades those structural cells to begin multiplying.
Yes, exactly.
But I'm struggling with the mechanics of what happens next. If the tissue is severely inflamed, the immune alarms are blaring, and antibodies are flooding the area. How does the bacteria survive long enough to keep spreading? Those seem like contradictory states.
They do. But the bacteria utilizes a brilliant stealth mechanism to solve that exact problem. Once inside an epithelial cell, it expresses a surface protein called IgSA.
Sometimes referred to as VIRG, right?
Right. And this protein interacts with the host cell's internal structural scaffolding, the actin cytoskeleton.
Okay, so it messes with the cell skeleton.
Yeah, Shigella forces the host's actin to polymerize at one pole of the bacterium, weaving this dense structural tail.
It builds an actin rocket out of our own cellular architecture.
Literally an actin rocket,
Yeah.
propelling the bacterium forward at incredible speeds.
That is mind-blowing.
The actin comet tail pushes the bacterium through the cytoplasm right to the edge of the host cell membrane. And it pushes so hard, it forms a protrusion that punches deep into the neighboring epithelial cell.
Oh, so it just rams into the next cell over.
Yes. And the adjacent cell then pinches off that protrusion, absorbing the bacteria.
So it moves from cell A directly into cell B without ever entering the extracellular space.
Exactly. It spreads laterally while remaining entirely intracellular. The host's humoral immune system, the antibodies and complement proteins circulating the fluid, they cannot neutralize what they cannot see.
It is completely invisible to them. And the convergence of this evolutionary trick is just nuts. The notes point out that Shigella isn't the only pathogen to figure this out.
No. Unrelated pathogens arrived at the exact same molecular solution for intracellular stealth. Listeria monocytogenes, which is a completely different class of bacteria, utilizes an actin comet tail.
Wow.
And the vaccinia virus, a massive pox virus, does the exact same thing.
So you have entirely different kingdoms of life independently solving the physics of intracellular movement through the exact same mechanism of actin polymerization.
It's convergent evolution at its finest.
That is wild. But, witnessing a pathogen with this level of stealth tear through a vulnerable population forces a critical look at the human stakes. The 1994 Rwandan refugee crisis in Goma really demonstrates the catastrophic potential of this biology.
It does. It's a tragic example. During that crisis, over 1 million refugees fled across the border into Zaire. They established camps with just unimaginable population density.
And the sanitation infrastructure was probably non-existent.
Non-existent, and access to clean water was severely limited.
Which is a perfect storm for a pathogen that requires only 10 organisms to establish an infection.
Right. An outbreak of Shigella dysenteriae type 1 ignited in those camps. And the resulting mortality was staggering, with case fatality rates in certain areas reaching 30 to 40%.
30 to 40%. That is an extraordinarily high lethality for a diarrheal disease.
It was horrific. And it wasn't just the fluid loss. It was a brutal convergence of profound underlying malnutrition, a near-total absence of supportive medical care, and the systemic damage caused by the Shigella toxin.
Right, because the toxin enters the bloodstream and causes kidney failure.
Exactly. It's a multi-system failure at that point.
Which makes the clinical response to this disease so critical. Like, if someone develops severe cramping diarrhea, the immediate instinct is often to reach for an over-the-counter anti-diarrheal medication.
Sure, like Imodium.
Yeah, but doing that with invasive dysentery can literally be fatal.
Antimotility drugs, like loperamide, function by paralyzing the smooth muscle of the gut to slow down transit time.
Right, to stop you from having to run to the bathroom.
Right. If a patient suffering from simple secretory traveler's diarrhea, slowing the gut provides symptomatic relief, but applying that same logic to an invasive pathogen creates a severe clinical hazard.
Because you are locking the Trojan horse inside the city.
Exactly. By slowing intestinal motility, the host is artificially trapping the invading bacteria and their destructive toxins against the mucosal wall for an extended period.
Giving it more time to just burrow in.
Yeah, it provides the pathogen more time to burrow into the tissue, significantly worsening the necrosis and increasing the risk of systemic complications, including toxic megacolon.
Oh, God. So when there is blood in the stool, attempting to stop the gut from flushing the pathogen is incredibly dangerous.
It's one of the biggest warnings we give. Do not use loperamide for invasive dysentery.
Good to know. Now today, a PCR test can identify the genetic signature of Shigella almost instantly, alerting a doctor to avoid loperamide. But before DNA sequencing, how did researchers actually identify this invisible cellular hijacker? Relying on symptoms alone wouldn't confirm this specific pathogen.
Right. Well, before genetic markers were available, the scientific community relied on the Serenity test, which was developed in 1955.
The Serenity test, what did that involve?
Researchers would culture the suspected bacteria and apply a drop directly to the eye of a guinea pig.
Wait, the eye? Why the eye instead of just observing it in a petri dish or, I don't know, feeding it to the animal?
I know, it sounds bizarre. But if the strain carried the PINV plasmid and was genuinely invasive, it would penetrate the tissue of the eye, causing a severe pus-filled infection known as purulent keratoconjunctivitis.
Okay, but why use the eye specifically?
Because the cornea is a unique tissue. It's avascular, meaning it lacks regular blood flow.
Ah, so it's shielded from the immune system.
Exactly. It's largely shielded from the systemic immune system. This provided researchers a living cellular matrix to observe pure, unchecked bacterial invasion, without the animal's circulating antibodies immediately neutralizing the threat.
Wow.
It was an undeniably crude metric, but for decades, it was the gold standard to separate true dysentery-causing strains from non-invasive environmental bacteria.
That is fascinating. Thankfully, modern diagnostics rely on molecular assays now. But even with advanced detection, treating Shigella is becoming an escalating crisis. The pathogen is out-evolving our pharmaceutical arsenal, isn't it?
It really is. Shigella has systematically acquired resistance to nearly every first-line class of antibiotics.
It's burning through our options.
Yeah, in the 1980s and 90s, widespread resistance to ampicillin and sulfa drugs emerged. Since the early 2000s, resistance to fluoroquinolones like ciprofloxacin has basically become a global standard.
And currently, the CDC is intensely tracking extensively drug-resistant, or XDR, strains of Shigella.
Yes, XDR strains are a major concern right now.
Strains that are simultaneously shrugging off all the heavy hitters.
We are seeing isolates resistant to ciprofloxacin, azithromycin, and ceftriaxone all at once. And these XDR strains are rapidly circulating among international travelers and within sexual networks.
And that loss of antibiotics is reviving the intense debate over vaccine development, right?
Yes. Historically, a Shigella vaccine was deemed a lower commercial priority because the assumption was always that cheap antibiotics would just work.
But today, the lack of a broadly licensed vaccine is a glaring vulnerability in global health security.
That's a huge blind spot.
Which sort of circles back to the biological lie we started with. Shigella is genomically just E. coli with specialized armor.
Right.
Yet the medical utility of keeping the name Shigella is absolute, isn't it? Like, if a diagnostic report identifies E. coli, a physician might default to treating a standard urinary tract infection or self-limiting food poisoning.
Exactly. But the word Shigella triggers an immediate, specific clinical pathway.
It alerts them to the danger.
It alerts the physician to the low infectious dose, prompting immediate isolation protocols. It explicitly warns against prescribing antimotility agents like loperamide. And it directs the team toward highly specific antibiotic stewardship to counter that XDR threat.
So in this case, the necessity of saving lives and preventing explosive outbreaks completely overrides the rules of evolutionary taxonomy.
Medical utility wins every time.
It makes perfect sense. So let's pull all of this together. we've explored a pathogen that operates as the ultimate microbial infiltrator.
It really does.
Shigella utilizes a minuscule infectious dose to survive the stomach, actively exploits our immune surveillance to breach the gut lining, injects a molecular syringe to trigger a fiery macrophage explosion, and builds actin rockets from our own cellular scaffolding to spread invisibly.
And the ongoing human toll requires our attention. The 2013 Global Enteric Multicenter Study, known as GMS, which was published in The Lancet, provided a really sobering look at this pathogen's reach across populations in Africa and Asia.
What did they find?
The study identified Shigella as one of the very top causes of moderate to severe diarrhea in young children.
Which is devastating.
It is, because beyond the acute threat of mortality, these early childhood infections inflict severe, repeated damage to the gut architecture.
Ah, so it causes long-term issues.
Yes. This leads to chronic malabsorption of nutrients, resulting in long-term malnutrition and permanently stunted cognitive and physical growth.
So it's a 19th century disease still dictating the health outcomes of the 21st century.
Exactly.
But, diving into these sources reveals an even more unsettling reality about how these microscopic weapons evolve.
Oh, yeah.
We discussed the PINV plasmid, the genetic blueprint for the invasion toolkit. But microbes do not keep their blueprints proprietary, do they?
No, they don't. The sources note that Shigella shares its type 3 secretion system, that molecular syringe we talked about, with entirely different pathogens, like Salmonella.
Because of horizontal gene transfer?
Right. Horizontal gene transfer is the engine of bacterial evolution.
Mm-hmm.
They readily swap these genetic weapons schematics.
Which brings us to the deadly Shigella toxin. Historically, the genetic instructions for that toxin were passed horizontally via a bacteriophage, a virus that infects bacteria directly to other strains of E. coli.
Yes. And that singular transfer event gave rise to the infamous E. coli 0157.H7, which is the strain that causes severe kidney failure and contaminated food outbreaks.
So OV virus acted as a courier, basically delivering the most lethal component of Shigella to a completely different bacterial population.
Exactly.
Consider the implications of that mechanism for a second. If these bacteria can seamlessly upload and download genetic blueprints for cellular sabotage, what happens as these new, extensively drug-resistant Shigella strains mingle with the trillions of other bacteria residing in the human gut?
It's a terrifying thought.
It is a stark reminder that the microbial arms race is not confined to laboratory petri dishes. It is happening continuously inside us. The evolutionary mechanics are still down there in the dark, bolting new armor onto the family car.